Okay, next we will look at the mycoplasmas. Now these are the tiniest, smallest, tiniest, minusculest, smallest prokaryotes, replicating prokaryotes. Okay, so the smallest of all is your mycoplasma. The oldest of all is your archaebacteria, no? So mycoplasmas, they are the smallest independently replicating prokaryotes. Okay, independently replicating because they are not like virus, no, where they need to attack a cell and replicate within. They can replicate on their own. They were discovered by Pasteur when he studied the bovine pleuropneumonia. Okay, they were discovered by Pasteur. Then the causal agent of this bovine pleuropneumonia was cultured by Knockaert and Roux, and the generic name, means the genus of Mycoplasma, was given by Novak because they have a fungus-like resemblance. Okay, mind you, these are not fungus. They look like fungus, but they are not. They just look like. Okay. Now they are parasitic as well as saprophytic. So they can live on decaying matter as well as they can live on other organisms. There are around more than 200 mycoplasma-like bodies that are associated with sewage, plants, animals, insects, hot water springs and other high temperature environments. They are found in the phloem tissues of diseased plants. Okay. So they can live in so many places. They can live in the sewage, in the plants, in the phloem, in the insects, in the animals. And in humans also, we find them at least 11 different types of mycoplasmas we can find in humans. Mycoplasma orale, Mycoplasma salivarium. Okay, and this is found in every adult. Even you have, even I have this one in my saliva. I also have, you also have Mycoplasma salivarium. Okay, so let's look at the general characters. They are single-celled, so unicellular. Smallest, not just small, they are the smallest. They cannot move, non-motile, and obviously they are prokaryotic. Okay, they form an fried egg-shaped colonies. Okay, it looks like it's when you fry an egg. Today, suppose you fry an egg. No, you just see and you think, oh, I'm going to eat mycoplasma in with my Mycoplasma salivarium. So that you remember and you feel disgusted, so you won't eat egg. Okay, so these are the shapes, the diagrams. You can see the colony form, then you can see entire colonies, then you can see the longitudinal section, and you can see the spherical form and the irregular filamentous form. Okay, so those are it's all there in the diagram, you see. Okay. Now let's look at some forms. They are pleomorphic, means, let's continue with the characters. They are pleomorphic, means they can change their shape depending upon what culture media you keep them on. Okay, suppose you want to grow them in one type of media, they will have a different form. In another different media, a different form. They may be rod-like, ring-like, like a globe or like filaments. So you saw in the diagram. They assume mostly, though, they prefer to have a spherical shape. Most of them have almost round shape. They are very much small, so they can pass through almost all the filters. Okay, they are ultra filterable because they are so small. Okay, so like when you wear a mask, no? Now because of COVID, you're wearing a mask, 3-ply mask. They have 3 fibers that keep getting smaller and smaller. Like the first layer, no? These fibers are, let's say, larger. Then the second layer of mask will have smaller fibers. Then the last layer of mask will have even smaller fibers, so that when the virus comes, it might get stuck here or there, or at least it will get stuck in the third one. No, that's why you wear 3-ply mask. So if you filter, okay, this is the filter. If you keep as many filter papers as possible, which are very fine pores, still the mycoplasma can pass through because they are so small. Okay, they will just seep through because they are so small. So they do not have a rigid cell wall. Okay, they are triple layered lipoprotein. So they have fat and protein in their cells. So inside, let's look at the inside part, the internal structure. They have cytoplasm and ribosomes. They are found scattered in the peripheral zone. Okay, I think better we just look at the image first. Okay, so you can see this diagram. Let's look at the figure name. Figure name is mycoplasma, structure of a typical cell. Now, okay, I think that's a better view. So you can see the lipoprotein membrane. Okay, so this membrane is made up of fats and proteins. Okay, lipid means fats and proteins. Then you are having the cytoplasm, lots of cytoplasm. Okay, and in the cytoplasm we are finding ribosomes in the around the outside layer, so around the periphery. Okay, around the periphery. In the center only one was there, maybe, but not mostly in the center. We are finding mostly in the periphery. Then you are finding some DNA material. Okay, obviously there should be DNA, otherwise how you can replicate. Then there are some soluble proteins and some metabolites. Okay, now let us see what they have written. Okay, so the ribosomes, they are 14 nanometer in diameter. Here I'm reading here. And they resemble the bacteria in sedimentation characteristic. Okay, the ribosomes are of the 72S type. And inside the cytoplasm we find fine fibrillar DNA. Okay, a very thin DNA. But it is double-stranded like this. So if you look at the image, you will notice that it is double-stranded. No, this one like this, it's double-stranded. Okay, but it's a very fine fibrillar double-stranded DNA. They grow, they are not as fast in growing as bacteria, and they need sterol for their nutrition. Sterol is sort of a fat, it's a steroid, no? Oh, it's some sort of a hormone, I think. So they are resistant to antibiotics, but they are sensitive to tetracycline. So you cannot kill these with antibiotics like penicillin, this one, etc. But they can be killed by tetracycline and also by temperature of 40 to 55 degrees Celsius for 15 minutes. Okay, that's why you boil your water, no? That's why we don't like add antibiotic into water and drink it. We don't do that. We boil our water, no? Because it kills almost all the microbes. I think it kills all. No, as though we boil like 100 degrees, right? So they do not produce spores and they divide by binary fission. So few points you can remember is enough. They divide by binary fission. They are killed by high temperature. They don't produce spores. They are resistant to certain antibiotics but sensitive to tetracycline. They have a fine fibrillar DNA which is double-stranded. Okay, the ribosomes are 72S type and they are found scattered in the peripheral zone. Okay, and the membrane is lipoprotein layer. So more than enough, I think. Okay, so basically this is almost the same. The size is 300 to 800 nanometer in diameter. They have a thick lipoprotein layer. I already told you that. The ribosomes are present. DNA is present. 72S type, double-stranded helix DNA. We have already done that. Okay, there's one new. The RNA is about 8%. Okay, the DNA is about 4% and RNA is about 8%. and is less than half of the other protoplasms. What does that mean? Or because the DNA is very small, no, it's very fine. So just 4% of the entire cell is DNA. But there are more RNAs, no, because the ribosome is quite large. Okay. So example is your Mycoplasma aureale, then salivarium. So with this we are done. Let me just upload this one.